Thank you for that. Thank you for that kind introduction, Eduardo. It is my pleasure to be here and to present the research we have been performing in my laboratory during the last few years. And I'm going to try now to share the screen to have my presentation. Let's see if everybody can
Yes, we can see it.
see, but I'm going to go into presentation style now. And then try to move this.
Yeah, we had that issue before.
And then let me see if I can. Well,
Yeah.
actually, can you see the cursor or not?
Your cursor? No, I cannot see it.
Which screen do you see? Do you see the?
We've seen a bigger screen, a small screen on the side, and at the bottom of the slide. That's what you see on your screen. Somehow, this environment, we had that problem before. It looks like you need to get off of the presentation mode and present because you have the bigger image. Yeah, it's much bigger. Yeah, and go from there.
Okay, let's do it this way then. Let me just move it so that you see it in front of
Oh,
you.
Dr. Aladab is saying just from the top left, you can click swap screens. Oh,
Yeah, that's what I did. But then what I realized is that then then you were not seeing the screen that I thought you were gonna see. Um,
yeah, it's
Let me see. Oh,
okay. Now it's good.
yeah, so let's do it this way. But then another thing is, do you see the cursor or not?
I can see your cursor. Yeah.
Okay, let's do with that cursor instead of the laser point. So thank you very much again for the introduction. So my name is Luis Martinez. I'm going to talk about mechanisms of arterial stiffening in obesity and diabetes. And I want to first say that one of the major aims of my laboratory is to identify the mechanisms that cause arterial stiffening, in particular in conditions of obesity and diabetes, hypertension, and aging. And the goal is to develop interventions, whether it's exercise, diet, or pharmacological therapies, to try to reduce vascular disease and associated problems with it, such as peripheral artery disease, cardiovascular, cerebrovascular disease, aneurysms, and other conditions. Now, one of the things that I'm very proud of is to say that I form part of a group that we call Vasorum. We performed what is called team science. So the results of the research that I'm going to present today is actually the result of this team that is composed of Drs. Jaume Padilla, Dr. Camila Manrique, and myself. We call ourselves Vasorum, the Vasorum team. And part of our research is focused on arterial disease and arterial stiffening. Now, with that being said, let us get into what is arterial stiffening and its connection to cardiovascular disease. I want to mention here that I often see the term arterial stiffness being used, and I want to remind everybody that stiffness is just a characteristic of a material, the same as temperature is also a characteristic. So temperature can be hot or cold, stiffness can be high or low. So when we talk about arterial stiffening is the process of increasing arterial stiffness in blood vessels, and what is its association to cardiovascular disease. So if we were to take a blood vessel isolated and increase intraluminal pressure under passive conditions, that is avoiding the constriction of the vessel in the form of aortic constriction of a smooth muscle, then we would see that the vessel, in response to the increasing pressure, distends and increases its internal diameter. So a stiffer vessel is a vessel that does not distend as much as a softer or more elastic vessel. So how is that physiologically incorporated into a whole system in which the heart is pumping blood. So during systole, the heart pumps blood, and that blood that comes out from the heart into the aorta increases intraluminal pressure that distends the artery. When it is an elastic artery, then it distends properly. And then during diastole, the vessel recoils and then pushes that blood out of the aorta, also increasing diastolic pressure. And therefore, since there is a reduction or dampening of the systolic pressure as the vessel distends, and then an increase in diastolic pressure as the vessel recoils, the pulse pressure is relatively small. But then when the artery gets stiffer, there is no distension. This increases systolic pressure because the pressure that is initiated by the pumping of the blood from the heart cannot distend the artery. That increases systolic pressure, and that reduces diastolic pressure because there is not that recoil of the aorta, therefore increasing pulse pressure. So that is what happens when the aorta is stiffer. And one of the most important things to consider is that an increase in aortic or arterial stiffness is an independent risk factor for cardiovascular disease, including the incidence of heart attacks and stroke. What is the way that we use in humans for non-invasive measurement of arterial stiffness? And the gold standard is the calculation of pulse wave velocity. And pulse wave velocity is the measurement of the distension of the blood vessel that occurs as measured with an ultrasound probe in the carotid and also in the femoral artery. And then we can measure the carotid to femoral artery pulse wave velocity. So that is how fast the wave of pressure or distension of the wall in the arteries goes, how fast it goes from the carotid to the femoral. And we can measure the distance and we can measure the velocity of that wave form, and that tells us what is the pulse wave velocity and is associated with the stiffness of the blood vessel. In this case, it is important to mention that because it is in vivo, it is influenced by several factors, including, for example, blood pressure. Now, in research, we can also measure pulse wave velocity in a mouse by putting an ultrasound probe and getting B and M mode images over time. And we can measure the distension of the wall of the aorta and then how that distension travels along the aorta, along a known distance in the aorta, and we can measure then a pulse wave velocity. An increase in pulse wave velocity means that the artery is stiffer. A reduction in pulse wave velocity, that means that the artery is softer. Now, what is the pathophysiology and the importance of this arterial stiffening or increase in stiffness of the blood vessels, in particular on the arterial side? So if we have a compliant aorta on this side, we can see that the pulse wave travels along the aorta downstream, and then as it travels and it gets into the bifurcation of the iliacs, this pulse pressure actually returns, and then it allows for better perfusion of the heart through the coronary arteries. So when the aorta is pliable and compliant, then that causes for the systolic blood pressure to be normal, and then for diastolic blood pressure to be also normal, and then we have a certain level of pulse pressure. And then this favors the timing of the wave reflection, increases coronary perfusion, and the pulsatility of the blood is lower as it goes into the smaller vessels and the microcirculation. When the aorta is stiffer, then there is a fast pulse wave velocity that returns too fast to the heart and does not allow for proper coronary perfusion. It increases systolic blood pressure and decreases diastolic pressure, increasing pulse pressure. And that pulsatility is transmitted to organs, and then it can cause end organ damage, in particular to those organs that receive a good amount of blood, such as the kidney, the brain, and the womb. Now, one thing that is very important to consider is that for a long time, it was considered that arterial stiffening was a consequence of hypertension and also a consequence of aging. But then there was a seminal paper published in Hypertension in 2013, in which it showed that arterial stiffening precedes systolic hypertension in diet-induced obesity in mice. And the paradigm, which is still current and many people address to it, is that the major cause of arterial stiffening is changes in the extracellular matrix, particularly an increased presence of collagen that causes a reduced luminal diameter because the vessel is not able to distend as much as an elastic artery. And then that there is also elastin fragmentation. This is what I call the extracellular matrix paradigm, which is true to a certain extent. But I'm going to try to convince you that there are other pathways involved in arterial stiffening, particularly in obesity and diabetes. Let me wait until this noise goes away. Good. Now, one of the things, some of the ways in which we use to measure these changes in extracellular matrix when we can isolate blood vessels from either animal models or even from human subjects is to do Mason's trichrome staining to stain for collagen. We can do also second harmonic imaging to obtain images of collagen without staining blood vessels, or we can use also precocilious red, which is another stain that detects collagen accumulation. For elastin, we usually use VVG staining that stains elastin fibers, or we can use a hydrazide to stain the elastin fibers, in which in this small vessel, we can see the internal and external elastic lamina that are full of elastin in this blood vessel, or we can use autofluorescence. Now, I'm going to present to you data that indicates that particularly in obesity, we see stiffening of the smooth muscle cells, which are conform the medial layer of the arteries, and that this stiffening of the cells is caused by an increased presence of stress fibers or F-actin within the smooth muscle cells of the blood vessel. And then with this stiffening, as I mentioned before, it causes the vessel to be less distensible in response to increases in intraluminal pressure compared to a more elastic artery. This causes a shift to the left in the strain-stress relationship. That is, the vessel is not able to distend as much, and that increases the modulus of elasticity. An increased modulus of elasticity means that the vessel is stiffer. And I'm going to show you data that indicates that these vessels that are stiffer, particularly in animal models and subjects with obesity and type 2 diabetes, have an increased presence of stress fibers in the smooth muscle cell. This is a stain of F-actin fibers in a blood vessel, and you can see the amount of stress fibers that are present in the smooth muscle cells. So I'm going to show you data that indicates that vascular smooth muscle cell fibers, stress fibers, are increased in stiffer arteries that causes vascular stiffness. And of course, this is associated with an increased cardiovascular risk. Now, in the cytoskeletal paradigm shift that we are proposing that has been taking place over the last few years, almost probably 10 years or so, is that stiffer arteries are characterized by presence of endothelial dysfunction. Presence of endothelial dysfunction means that there is a reduced nitric oxide bioavailability. And this the most physiological parameters that produce or events that produce nitric oxide are flow-mediated dilation and insulin-induced dilation. This causes the stiffness or increases the stiffness of the blood vessel, reduced luminal diameter, and we see an increase in F-actin formation, an increase in activity of an enzyme that is called tissue transglutaminase, and of course we see cellular stiffness in addition to an increased expression of the receptors for the extracellular matrix collagen or other extracellular matrices, the integrins alpha B beta 3 and alpha 5 beta 1. Now, we have been for a while proposing the existence of a signaling corridor that associates endothelial dysfunction, a reduction in flow-mediated dilation and insulin-induced dilation, with increased TG2 activity, F-actin formation, and cellular stiffness that ends up in arteries being stiffer. So I'm going to present this signaling corridor that I'm talking about. This slide is a little busy, but I'm going to first show it to you and then repeat the individual steps as we go through the data, through the results of our research. So one of the things that we have seen is that in the endothelium is, it has a structure in the luminal side that protrudes into the lumen that is called the glycocalyx. And here is an electron micrograph of the glycocalyx of a blood vessel. And this structure is conformed of glycoproteins, glycosaminoglycans, proteoglycans. This is called the glycocalyx. And as you can tell, since it's facing the lumen, it can detect flow, is a mechanosensor of shear stress. And as it detects flow, it produces nitric oxide and induces vasodilation. One of the first things that we found is that in animal models that had obesity and diabetes, we saw an increase in an enzyme present in the plasma that is called neuraminidase. And neuraminidase is an enzyme that desialylates components of the glycocalyx. And then if we stain the glycocalyx with germ agglutinin, we can see that a control artery is well covered with this glycocalyx structure. But when it's exposed to neuraminidase, we destroy that glycocalyx structure. So we show that destruction of this glycocalyx reduces the capacity of blood vessels to detect shear stress induced by the flow of blood, reduces nitric oxide production, and therefore reduces vasodilation. At the same time, nitric oxide is an endogenous inhibitor of the enzyme tissue transglutaminase, or TG2. And TG2 is an enzyme that has several functions. One of them is that it activates the ROC limb kinase pathway. And by activating this limb kinase pathway, it phosphorylates cophylline. Cophylline is in charge of severing actin fibers. So when cophylline is deactivated, then that increases actin polymerization, formation of F-actin stress fibers, cross-linking of these fibers, and stiffening of the smooth muscle cells. So therefore, when we have low nitric oxide, which is the endogenous inhibitor of TG2, we have increased this pathway and we have increased stiffness of the cells and of the blood vessel. In addition, I told you the other physiological means by which the endothelium produces nitric oxide is through insulin stimulation of the insulin receptor in endothelial cells that induces insulin signaling and production of nitric oxide. And I'll show you data that neuraminidase also reduces the presence of this insulin receptor in endothelial cells, therefore reducing the capacity of insulin to induce vasodilation and produce nitric oxide, which again, a reduction in nitric oxide bioavailability allows for the increased activity of TG2 and the stiffening of the smooth muscle cells. I know it's a lot to take for now in this corridor, but I'll show it to you step by step as we go through the data. So the first thing that I told you we found is that in mice that were fed a Western diet for 16 weeks, we found that those mice had increased plasma neuraminidase activity. And I told you plasma neuraminidase sheds glycocalyx structures, inducing glycocalyx degradation and reducing the capacity of these glycocalyx structures to sense blood flow and shear stress. We corroborated this in humans in that when we compared type 2 diabetic humans with healthy controls, we see that they also have increased plasma neuraminidase activity. They have also reduced plasma nitrite, which is a surrogate of nitric oxide. So therefore, they have reduced nitric oxide bioavailability. And they also have reduced flow-mediated dilation as collected from the femoral artery. So we have this correlation between these results in humans as well. Now, if we expose mice to neuraminidase, We using atomic force microscopy, which is a cantilever that touches the surface of the endothelium. We can detect the length of the endothelium, and we see that injection of neuraminidase reduces the length of the glycocalyx on the endothelium. And then if we perform in isolated arteries that were exposed to neuraminidase flow-mediated dilation, we see that their dilation in response to flow is reduced compared to untreated vessels. This is shown in response to flow and in response to shear stress. But when we look at the dilation of the vessels in response to acetylcholine, which is a receptor-mediated dilation, there is no differences. And when we bypass the endothelium and we induce dilation with sodium nitroprusside, which is a nitric oxide donor or mimetic, we see that there is no changes between the arteries that were treated with neuraminidase or the controlled ones, indicating that the destruction of the glycocalyx was what reduced the response of the vessels to flow-induced dilation. So we asked the question, can we improve flow-mediated dilation in diabetic mice by inhibiting neuraminidase? So there are in the market FDA-approved drugs that reduce neuraminidase activity. One of them is zanamivir, which is Relenza, that is used to treat influenza. And one of the first things we did was, can we give inhaled zanamivir as it's prescribed in humans to mice and increase plasma zanamivir in these mice? And we saw that if we give zanamivir to mice via inhalation, we can increase plasma zanamivir presence. Then we measure plasma neuraminidase activity in DBDB mice, diabetic mice, that were treated with zanamivir for 5 days. And we see that we can significantly reduce plasma neuraminidase activity, and that we also were able to reduce aortic stiffness as detected by AFM. We can increase glycocalyx length after the treatment with zanamivir. And because it was a short time, that's what we believe, it was a short time treatment, we didn't see any changes in pulse wave velocity. But indeed, we are influencing the characteristics of the glycocalyx. Then we isolated mesenteric vessels and then perform flow-mediated dilation. And we see that in the DBDB diabetic mice, flow-mediated dilation is almost non-existent, but we can restore that dilation when we treat the mice with zanamivir, not to the level of the non-diabetic mice, but we can substantially improve flow-mediated dilation. Interestingly, we can also improve acetylcholine-induced dilation and also insulin-induced dilation, especially at very high doses of insulin, without affecting the endothelium-independent vasodilation to sodium nitroprusside. So this impelled us to try to do a clinical trial in which we gave diabetic patients zanamivir for 5 days, as it is prescribed for influenza treatment. And we were able to increase plasma zanamivir presence after the treatment, but only modestly. Unfortunately, this increase in plasma zanamivir was not able to significantly reduce plasma neuraminidase activity and was not able to change the characteristics of the glycocalyx or improve flow-mediated dilation. At this time, it is our hypothesis that because we did not achieve sufficient concentration of plasma zanamivir, we were not able to restore the glycocalyx, but that remains to be determined. Now, switching to the corridor that links shedding of the insulin receptor on the endothelial surface with reduction of nitric oxide and then activation of TG2. But first, let's look at this specific signaling pathway. We previously have shown that ADAM17, which is a metalloproteinase that is attached to the cell membrane, can cleave or shed the extracellular portion of the insulin receptor, which is the alpha subunit of the insulin receptor. And we show now that neuraminidase activity can activate ADAM17, which in turn sheds the insulin receptor. And that reduces the capacity of endothelial cells to produce nitric oxide in response to insulin stimulation. The first thing that we observed was that when we obtain samples from the jejunum of individuals undergoing bariatric surgery, we isolated jejunal arteries, and we determined their vasoactive characteristics. First, we perform a concentration response curve to phenylephrine, and we show that both arteries from non-diabetic and diabetic subjects constrict to the same extent in response to phenylephrine. They also dilate to the same extent to acetylcholine, but we saw that they do not dilate the same to insulin increasing concentrations. The insulin-induced dilation in the diabetic patients is dampened. There are no differences if we bypass the endothelium and induce relaxation in response to the nitric oxide donor sodium nitroprusside. So this told us that there were some problems in the insulin signaling pathway in endothelial cells to produce nitric oxide. So the first thing that we did was to look at the insulin receptor. The insulin receptor contains two subunits, the alpha subunit and the beta subunit. The alpha subunit is extracellular, and the beta subunit is transmembrane and intracellular. And we found that in type 2 diabetic subjects, the amount of insulin receptor alpha is reduced in all of the components of the vascular wall, the intima, that means the endothelium, the media, and the adventitia. Whereas the insulin receptor beta, which is the transmembrane and intracellular portion of the insulin receptor, there is no change or no difference between non-diabetic and diabetic subjects, which suggests to us indeed that the insulin receptor was being shed. Then we decided to look at some of the enzymes that could be shedding the insulin receptor from endothelial cells, and receiving some advice from other investigators at the University of Missouri, we decided to look at ADAM17, which is a metalloproteinase anchored to the cell membrane that is a famous sheddase. He's very famous for shedding tumor necrosis factor alpha, TNF-alpha. And we found that indeed, in the wall of the arteries of diabetic subjects, there was an increase in ADAM17 compared to those non-diabetics as normalized to beta-actin. We also look at the presence of TIM3, which is an endogenous inhibitor of ADAM17, and we found that the amount of TIM3 was reduced in diabetic subjects, which suggests that there is an increased activity of ADAM17 in the wall of the arteries of diabetic subjects. We then look at some characteristics of the activity of ADAM17 in human subjects. And as I told you before, we did another cohort of individuals with type 2 diabetics and then compared them with healthy subjects. And we saw again that plasma neuraminidase activity is increased in the diabetic subjects. And we also saw that in those same subjects, there is an increased activity of ADAM17 in plasma. Then when we did an oral glucose test that increases insulin in circulation, we see that there was an increase in leg blood flow after the oral glucose test, suggesting that there is insulin-induced vasodilation, and that this insulin-induced vasodilation is dampened in type 2 diabetic subjects. When we did vascular conductance, which normalizes this to changes in blood pressure, we again saw that the difference remained. The individuals with type 2 diabetics had a reduced vasodilatory capacity in response to endogenous insulin after an oral glucose infusion. We then went mechanistically and tried to corroborate these findings in cell culture. So we culture endothelial cells, and then we exposed them to neuraminidase. And first, we corroborated that in this neuraminidase shed sialic acid from the surface of the endothelial cells. And we found that neuraminidase exposure to endothelial cells increased intracellular calcium, and that this increase in intracellular calcium caused an externalization of phosphatidylserine. And phosphatidylserine was also associated, the increase in phosphatidylserine was also associated with an increased ADAM17 activity in these endothelial cells. We also saw that in response to neuraminidase exposure, these endothelial cells lost insulin receptor alpha from their surface, and that they also reduce the phosphorylation of AKT, which is associated with insulin signaling. More than that, in isolated mesenteric arteries from mice, we found that arteries that were exposed to neuraminidase also had a reduction in insulin-induced dilation compared to arteries that were exposed to vehicle control, and that these arteries did not have any differences in dilation induced by exogenous nitric oxide as produced by increasing concentrations of sodium nitroprusside. So then, in this pathway of reduced insulin signaling due to the shedding of insulin, what we are proposing is that the exposure of neuraminidase induces an increase in calcium inside the cell, and that this activates an enzyme that is called NO6. It's a scramblase that moves phosphatidylserine from the inner leaflet of the plasma membrane to the outer leaflet of the plasma membrane. And it is known that flipping of phosphatidylserine to the external leaflet of the cell membrane activates ADAM17 or induces the bending of ADAM17 so that it can reach its substrates. And therefore, we believe that that is the pathway, or show that that is the pathway by which ADAM17 increases its shedding of the insulin receptor alpha. So to corroborate that, we expose cells to neuraminidase in the presence of absence of an NO6 inhibitor. And we see that when we apply the NO6 inhibitor, then we have reduced PS externalization, so the phosphatidylserine is not moved now from the inner leaflet to the outer leaflet. We see a reduction in ADAM17 activity. We see an increased presence of insulin receptor alpha in the surface of the endothelial cells, suggesting that the receptor is not being shed. And we saw an increase in AKT phosphorylation, which is the pathway of insulin-induced activity in endothelial cells. When we went ahead and we say, can we also block these effects of neuraminidase by blocking ADAM17 activity? So this is just to corroborate that indeed, when we put neuraminidase, we increase ADAM17 activity, that we can block that ADAM17 activity with an ADAM17 inhibitor, TAPIO, and that we block the insulin receptor shedding by including the presence of the ADAM17 inhibitor TAPIO. We have more insulin receptor alpha on the top of the cells. We improve insulin signaling with phosphorylated AKT, and we restore significantly, not to the levels of the untreated, but we restore significantly insulin-induced dilation without affecting the effect of nitric oxide on the dilation of these arteries. So I have shown you that there are several interventions that we can use to reduce endothelial dysfunction in type 2 diabetes. One of them is by blocking the activity of ADAM17 and reducing its shedding of the insulin receptor. We can also block the scramblase NO6 so that phosphatidylserine is not flipped to the outer leaflet of the membrane, and therefore ADAM17 is not activated. Or we can also block neuraminidase to block the initiation of this whole pathway that affects insulin-induced dilation, and of course also flow-mediated dilation. So now, I told you that the production of nitric oxide is an endogenous inhibitor of TG2, and that TG2, if it's not inhibited, it can then induce the production of F-actin stress fibers and the stiffening of the cells. So let's look at the evidence that we have on that regard. This begins with a seminal paper that was published in 2005, in which Eric Bakker and his collaborators found that tissue transglutaminase is needed for small artery remodeling and stiffening. Tissue transglutaminase, as I said, is an enzyme that cross-links glutamine residues with lysine residues, increasing the cross-linking of molecules, particularly extracellular matrix molecules, but as well as intracellular molecules such as actin, and that it also works as a G protein that can then induce the activation of the raw ROCK limb kinase pathway. And activation of this pathway, as I mentioned before, phosphorylates cophylline, inactivates cophylline, and therefore allows for actin polymerization to increase and increase the stiffness of smooth muscle cells. So we have data that show that if we expose isolated vessels that were cannulated to an activator of TG2, we increase transglutaminase activity, shown here by an increased fluorescence, and that the vessels that were exposed to the activator of TG2 remodel, and they are now stiffer and cannot achieve the same diameter as before under the same level of intraluminal pressure. If we block actin polymerization with cytochalasin D during this prolonged activation of TG2, we block this stiffening of the blood vessel and the remodeling. We also have shown that in small omeltar arteries isolated from individuals undergoing abdominal surgery, we see that if we compare the stiffness of those vessels between non-hypertensive and hypertensives, we see that they have a reduced distensibility or reduced capacity to be distended. We see a shift to the left on the strain-stress relationship, an increased modulus of elasticity, indicating that the arteries are stiffer. And also, we do several other different calculations. One is a calculated pulse wave velocity, and we see that these arteries are indeed also stiffer. Now, when we look at the structure of these blood vessels, we see that there is no significant changes in the amount of cells in the vessel wall, but there is an increase in the amount of phosphorylated cophylline. And I told you, when cophylline is phosphorylated, it is inactivated and allows for the polymerization of actin and formation of F-actin stress fibers. And indeed, we see in these vessels that there is an increased amount of F-actin stress fibers, and there are no changes in the amount of collagen as detected by second harmonic imaging. Now, we ask ourselves, can we block this pathway and prevent the stiffening of blood vessels? So we first tried an inhibitor of TG2, cysteamine. It's not a specific inhibitor because it also has other effects, such as an antioxidant effect, but it's an inhibitor of TG2. So what we did in this case, because we wanted to test its effect as a treatment, we fed female mice for 43 weeks to a Western diet. And then during the 8 weeks at the end of the feeding paradigm, we gave cysteamine in the drinking water to 10 of those mice. And then at the end of the trial, we measured pulse wave velocity, and it was followed by euthanasia and assessment of vascular function and mechanics. So what we found is that these mice that were fed a Western diet show a trend for an increase in mean arterial pressure that don't reach significant, and also a trend for a reduction on those who were treated with cysteamine. But when we looked at pulse wave velocity, we saw that mice that were fed a Western diet had stiffer arteries as determined by pulse wave velocity, and that cysteamine prevented or reversed that stiffening of the arterial circulation. And we also measure stiffness by atomic force microscopy by putting an end-phase sample of the aorta and testing the stiffness of the endothelium. And we saw that there was an increase in endothelial cell stiffness in the Western diet fed mice, and that it was reversed by treatment with histamine. Then we also isolated mesenteric arteries and looked at their distensibility by doing pressure diameter curves. And we obtained then the strain-stress relationship and the modulus of elasticity. And indeed, we saw that the arteries of mice that were fed a Western diet were stiffer, and that was reversed by addition of histamine in the drinking water during the last 8 weeks of Western diet feeding. Then we also tested this using a more specific inhibitor of TG2, which is a compound produced by the company Sedera. And we showed that the treatment with Sedera using a mini pump in DB diabetic mice did not change body weight, did not change blood glucose, but it reduced pulse wave velocity. Also, it reduced aortic wall stiffness as measured with atomic force microscopy, and also in isolated mesenteric arteries, it reduced the stiffness of these arteries as measured using pressure diameter changes under passive conditions. So I have shown you now data that endothelial dysfunction is associated with an increased activity of TG2 and stiffening of the smooth muscle cells that in turn participate, particularly in the initial stages, in the stiffening of blood vessels in diabetes and obesity. And that I would like to then at this time thank all the people who have participated in the research. There's many, as I told you, we are a team, the Vasorum team, that we perform studies looking at vascular function. And I would like also to thank the funding agencies, the National Institutes of Health, the American Heart Association, NSF, MU, of course, and other of the societies that have helped us perform this research. And I'll be happy to entertain any questions at this time. Thank you very much for your attention.
Thanks, Luis. This is a